Very small, nice audience here. My name is Lena Granovsky, and I'm leading the analytics and big data in Teva Pharmaceuticals. This is a use case studies session. So what I'm going to do in this presentation is to tell you a little bit about some of the things that we are doing in Teva and the interesting potential ideas that exists in pharma in general. So, we all know Teva is in the news today, so I decided to put this slide just to tell you some facts about Teva that might be interesting. So first of all, Teva was established in 1901, and today Teva is the largest producer of generic medicines. We actually leverage from over 1,800 different molecules in almost every therapeutic area. In addition to being the biggest generic producer, Teva works in specialty development, developing new drugs, and we're doing it actually in 4 therapeutic areas, mostly in respiratory, in headache and migraine, in pain and neurodegenerative diseases. We have we are active in 60 different markets, have 71 manufacturing sites, and have 43,000 employees. So just wanted to give you this information. Thought it might be interesting. After presenting Teva, I just want to put this disclaimer that everything that I'm going to talk now actually are on my views and not necessarily represent what we're doing in Teva and Teva's activities. Okay, so we all know that technology and artificial intelligence are here in the healthcare. People are being equipped with new monitoring devices, portables, wearables, implantables, digestibles, you name it. And we as patients, we are very ready to adopt these technologies. So we all are empowered with all the data that we're getting from the web, our apps on our cell phones. We are much more educated and less rely on our physicians to tell if we are sick or not. Physicians are almost ready, not as patients, but they're all also adopting the technology. We all know many interesting companies. Sorry. Okay, so physicians are also ready, and we all know about cool companies which helps physicians to make better decisions, diagnostics, mostly based on image and electronic medical records. But what about pharma? So there is an adoption of artificial intelligence and technology in pharma, but unfortunately the adoption of these technologies is much slower than one would imagine. And I think that here is the reason why. So I actually I came to Teva 2 and a little month ago, and I was really surprised to figure out that the process of drug development takes 10 to 15 years. So it takes 10 to 15 years to develop one drug. It costs more than $2.5 billion, and the success rates are extremely small. So out of almost 10,000 different components that start their journey here at the drug discovery, so the candidates, molecules that can be candidate drugs, only one ends up being an FDA and DMA approved drug, and it takes again 10 to 15 years. So I really believe that machine learning and artificial intelligence can be used, and big data can be used to improve each and every step of this process. So I will not talk a lot about different stages like drug discovery and preclinical, but I will concentrate on the clinical trials. So the clinical trials, this is something that takes 6 to 7 years. And it includes phase 1 studies, where phase 1 are small studies, usually in healthy volunteers. And the purpose of these studies is to show safety, just to show that the drug is not toxic and it's safe. And again, it's mostly done in healthy volunteers. So the phase 2 is actually proof of concept. It's also safety, but this is a proof of concept of efficacy. Does the drug work? And then the phase 3 study are the most massive studies with hundreds and thousands of patients, again to prove or disprove that the drug works and it is safe. Okay, so during these clinical studies, what would probably go wrong, right? It takes, by the way, I didn't tell you, but I understood why does it take such a long time? Why does it take 10 to 15 years to build a new drug? And this is because pharmaceutical company has to be 100% correct in everything she does. We cannot be 99.9% because this 0.1% or 0.1% can cost patient lives. So everything is so highly regulated and checked and is done step by step, just to make sure that nothing falls apart and we're, again, 100% sure that everything works. But still things happen, and a lot of things happen in the clinical studies, and these are some examples of what could happen. So first of all, how do we prove today that the drug works or does not work? So there is something which is called a clinical endpoint. Some measure which is defined in the protocol, and based on this measure there is a statistical test that compares, for example, migraine or amount of attacks, migraine attacks before the new drug and after the new drug. How do we know how many attacks did the patient have before and after the drug? So what patients usually do, they fill in electronic diaries or paper diaries or something. So they self-report on their condition. They are sometimes asked to report on their condition today, what happened to them a day before or 2 days before, and maybe a month ago. Who remembers what happened to them a month ago? When I ask, how did you feel? How were your pain levels during the last month? Or how was your sleep during the last month? I don't know to answer this question, but eventually this is how the efficacy of the drug is measured today, by these subjective reports of patients. The drug can work only for some subpopulation of patients, not for everyone. So it can be genetic differences, it can be different sex differences, and other factors that can impact if the drug works or doesn't work. We can take wrong assumptions. So like I said, there are statistical tests that are used at the end to decide if the drug works or does not work. So to do the statistical tests, we need some statistical assumptions. These assumptions are made from common knowledge, literature, physicians which have practices, they have this knowledge. But these assumptions are sometimes not correct, and sometimes the drug might work, but because of the assumptions we couldn't even prove if it worked or did not work. Data quality, operational failures. So sometimes the drug fails not because the molecule doesn't work, not because the pharmacology is not there, but the real reason for the drug to fail is actually the statistical analysis that we do. It's about what we are measuring and not how the drug works. And when this happens, I think for me at least, I see it's a huge opportunity. Because think of all these patients or severe patients with severe conditions that the drug, the molecule might have worked for them, but we just couldn't prove that it does. So, analytics and big data can help. So just an example of what can be done related to the previous slides. So the clinical trials, they need to be concentrated more on the patients. So first of all, better site selection. The clinical studies are done in physical sites. Physical sites is where the patient come to get the treatment, to get their drug. This is where their physician is. This is where they need to travel sometimes from a long distance, accompanied by family members many times. So choosing the best sites for clinical studies, this is a very important task, and this can be done using data. So we have historical data on different sites. We have internal data in the pharma companies about the performance of the sites. All this data can be used in order to better select sites. Better patient selection. Oops, sorry. Better patient selection. Again, we talked before that there might be a wrong patient population, so we can use data, electronic medical records, genomic data, proteomics data, different kinds of data, in order to better estimate if the drug will work for all the patients or only a subpopulation of patients. Real-time monitoring tools. So these clinical studies can take more than 1 year or 2 years sometimes to be completed. During these clinical studies, the patient sees the site or sees the doctor once in a while, once in 1 month, in a good case, sometimes once in 3 months. All the time in between, we are not gathering any information about this patient. So maybe his situation got worse. Maybe he deteriorated. So using, again, monitoring tools and analyzing the situation of the patient during the clinical study is also very important. And the most important thing, and this is what most of the efforts of today our group is in development of novel and personalized digital markers to be used as objective clinical endpoints. So as an example, let's say there is a drug for back pain. So the patient enters the study, and then he is asked, what are your pain levels? So he gives some level of pain, and then he becomes taking the drug. His situation gets better. He becomes much more active because of the drug. So the drug actually helps. But on the other hand, because the patient becomes much more active, his pain levels go higher. So after the trial, he comes again to the physician, and the physician asks again, okay, how are your pain levels? So his pain levels are actually the same as at the beginning of the study. So the conclusion would be that the drug doesn't work. But is it the case? Of course not. The drug works because he became much more active. So if we could measure the activity, for example, it's just one of the examples, and develop a personalized digital marker to prove or disprove if the drug works, it would really help. So this is only one example. And here is something that we do in the clinical study for cluster headache. So cluster headache is a very, very serious condition. It is a very, very strong headache. And this is one of the most painful conditions that the mankind knows. So it is rare, luckily for us. The pain is one-sided, and it is different from the headache. So the cluster headache has his own characteristics, which is different from the cluster headache. So what we are trying to do in this study is there is a drug that Teva develops for a cluster headache, and we try to build personalized digital markers in order to better measure patient condition. So the endpoint, how do we measure if the drug works or doesn't work? By electronic diaries. So the patient actually reports every day on what happened to him a day ago or 2 days ago. How many attacks did this patient have? These reports, from our experience and our knowledge, are very, very subjective and not correct. So what we did, we gave him an accelerometer watch. This is a medical device, a Philips accelerometer watch. And what we are getting, the data that we are getting, is actually data every 30 seconds. We're getting his activity level. And based on this activity level, we're trying to see if this patient had or did not have a cluster headache attack. So here are two examples. The first example is actually a nighttime attack. So it's probably hard to see, but it's 2 a.m. at night. And you can see, and the y-axis is actually his activity level, and the x-axis is time. So this is around 2 a.m. at night. You can see that this patient was mostly not active, but at some point he had these spikes of activity. And then this pink bar is actually what this patient reported in his electronic diary. So you can see that his report actually in this case was pretty accurate. The length is not exactly the same, but our algorithm was able to easily detect that here was an attack. And this is another example of the day attack. So here again, you can see the patient is active, but here he has a lower activity. The patient did report his attack over here. Again, a very good report. Sometimes the reports are totally off what we see with the real data. So they could be at a different day or different amount of attacks. So you can see here that that algorithm detected a cluster headache attack. But when we showed the data to the physician, he said, oh, you know what? It's probably not a cluster headache attack. I think it's just headache and not a cluster attack because the characteristic of cluster headache attack is different. So this data can also be used as a more precise measure, and the physicians can learn a lot by just looking into this data and having this much more accurate and objective measure for the drug. So this is one of the use cases. Of course, we use deep learning to do that. So now, after saying the keywords of deep learning, I want to show some more examples with actually smaller amount of data where other machine learning methods were used as well. So the example that I showed was the example in the clinical study. The next example is actually can be used both in clinical study setting, but also for disease management. And this is what actually today pharmaceutical companies need to do. They need to show not only the ability to develop a drug, but also show the benefits on the drug in the real life once the drug is actually on the market. So this is what is called beyond the pill solutions. So the next use case is a collaboration that we did with the Data Science Institute of IDC and the startup company called iFeel. The idea here is to collect passive data from patient smartphones in order to try and correlate it with the quality of life of patients and specifically with pain patients. So again, when the patient is in pain, his quality of life is not the best, and the way the patient reports on his quality of life is usually using paper or electronic diaries. So this is just an example of one of the questionnaires. So mobility, how was your mobility during the last month? What was the level of your discomfort during the last week? Were you depressed? Or things like that, that the patient come to the physicians once in a while, once in a couple of months, and they are asked these questions. Based on this, they are getting their therapist. So if we could monitor the patient again all the time, getting objective continuous measures, it would really, really help. So what we are trying to do here in this collaboration is to get different types of data from a cellular phone. we use the iFill platform. iFill is the startup company that we are using. We also, in addition to the passive data collection, which we will correlate with the measures of quality of life, we're also using voice application. So what this application does, it asks once a day a question, some question of pain patient. How did you feel today? And then it records the answer. And of course, we want to analyze this data, also the content and the semantic of the speech. Another use case is about Teva's digital inhaler. So a couple of months ago, Teva announced about the approval of his first digital inhaler. What digital inhaler is? So for patients who suffer from asthma or disease which is called COPD, Teva developed this inhaler, which includes a digital component in it. And what it does, it captures also the time of the inhalation. So every time the patient takes the inhalation, it captures the time. But also, in addition to that, and this is something proprietary for Teva, it also captures flow measures. So it captures how much volume did the patient inhale, how long did it take him to get to this maximum volume, measures like that, that can actually show if the drug was really delivered, and if the patient used the right technique in order to take this inhalation. Apparently, there is a huge unmet need in such a thing. Why? Because patients don't really remember if they did take or did not take their drug, first of all, and second of all, they don't know how to use the inhaler in the correct way, especially when they're in a hurry. They sometimes inhale really, really fast, and then they don't really get the real amount of the drug. So what we did, we actually wanted to build an algorithm based on the data from this digital inhaler in order to try and predict future asthma exacerbations. So we actually did it in 2 steps. The first one, we first used data from electronic medical records, only electronic medical records, to learn if we can choose features from the clinical data. So we worked with IBM Explorers, and we collected data on diagnosis, procedures, medications, labs, habits, and demographics of different patients. It is a big data set. It included about 3 million asthmatic patients. Built on this data, we figured out what are the clinical features which are relevant for asthma exacerbations. Once we have these features, we actually went to step 2, and we ran clinical studies. In these clinical studies, patients were given this smart inhaler, so the data was collected, and also that clinical data from the patient was collected together with their asthma exacerbations. So for every patient, we knew actually if it's a kind of time series data, and for each patient, we knew how many inhalations he took, when he took these inhalations, and the volume parameters of these inhalations. In addition to that, we also had these features from the previous data set and algorithm that we built. So here we had a small data set, only 65 exacerbation patients, and we had about even less than 400 patients in these studies. And again, clinical studies cost a lot of money, and this is why we not necessarily have huge amounts of data while building these algorithms. So we were able to learn a lot of interesting things. First of all, we were able to build a good predictive algorithm. The feature engineering and the feature selection piece was very, very important here. Again, we don't have a lot of data. And when we started first, we couldn't get to high precision metrics using this algorithm. So again, we learned the data deeply, and then we realized that we cannot build only one algorithm. We need to split these patients into two different subpopulations. So the first subpopulation was the patient who regularly used the inhaler, and the second one was the patients who did not use the inhaler regularly. So for those who don't use the inhaler regularly, the fact that they did use the inhaler was a good predictor. For the others who did use the inhaler in a regular way, the flow and volume information and other features which were collected from the digital inhaler were actually very, very important. So this improved the algorithm by significantly. So and these are some examples of the algorithm. So just to wrap up, I do think that there are a lot of interesting things, a huge amount of interesting things that can be done in pharma in each and every step of drug development. The biggest problem today is the regulatory problem, because these objective digital endpoints, they're not FDA regulated, they're not FDA approved. So if I want to measure activity and come to the regulator and say, okay, you know what, my drug worked because the activity was improved using my watch, this is not something that can be done today. So today, this is more an exploratory stage, and I really hope that this exploratory stage will become more and more regulatory validated, and this is something that is actually happening today with different consortiums, different pre-marketing pharmaceutical companies working together and building these tools. That's all. Thank you very much for your attention and not falling asleep at this hard time.